Thank you to the Oxford Vaccine Group for inviting me here to speak at your 10th immunisation seminar on vaccination against hepatitis B. So let's just recap briefly about what we know about hepatitis B. Approximately a third of the world's population have at one point in their lives been infected with the hepatitis B virus. That equates to about 2 billion individuals. And the World Health Organization estimates that worldwide there are 350 million people who are chronically infected with this virus. And about a quarter of these individuals will progress and develop severe liver disease. However, many people are asymptomatic, totally unaware of their infection. And worldwide there are over 1 million deaths as a result of the chronic complications of hepatitis B, which include cirrhosis and primary liver cancer. So hepatitis B is a global major public health problem. However, safe, effective and highly immunogenic vaccines have been available since the early 1980s. And in 1992, the World Health Organization recommended that all countries should introduce universal vaccination by 1997. So, for the historians amongst you, Hippocrates was the person who's been credited with firstly describing clinical hepatitis B infection in the 5th century BC. And it was mainly seen following military wars in the 17th and 18th and 19th century when people received blood transfusions and they developed jaundice following transfusions and long phase incubation. And it was termed serum jaundice. But one of the biggest outbreaks, which we now know is caused by hepatitis B virus, was seen in American troops in the Second World War who'd been posted overseas, went to North Africa. They received yellow fever vaccine, which is no doubt contaminated with infectious plasma, which caused over 26,000 cases of jaundice caused by hepatitis B, and the case fatality rate was 3 per 1,000 at that particular time. It wasn't until 1963 that Bloomberg, looking at the blood serum of an Australian Aborigine, identified what we now know to be the outer coating of the virus, hepatitis B surface antigen, and they actually termed that the Australia antigen. Later on in the early 70s, the actual particle was viewed using electron microscopy, and a few years later it was actually termed the Dane particle, and a few years later hepatitis B E antigen was identified. We now know hepatitis B antigen to be a marker of high infectiousness and high viral replication. And later on in the mid-70s in this country, screening for hepatitis B surface antigen began to prevent infected donations entering the pool of donated blood. And the discovery that antibody to hepatitis B surface antigen could protect individuals from acute and chronic infection after exposure led to the development of specific hepatitis B immunoglobulin, which is now used to confer passive immunity to individuals. And as I said, safe, effective immunogenic vaccines were available in the early 1980s. This is a diagram showing you the virus here. You can see the outer coating, the hepatitis B surface antigen, highly purified preparations of this antigen and the main component of the vaccine. 42 nanometers in diameter, consists of a partially double-stranded DNA. The core antigen is found within the liver. Hepatitis B core antigen is insoluble in the serum, only found in the liver. The antigen, as I mentioned, is a derivative of the core marker of high infectiousness, high viral replication. Now, so excess amounts of the hepatitis B surface antigen bud out into the serum and can be identified by using serological assays. And this is an electron micrograph showing you a blood pellet. The surface antigen exists as a spherical or tubular formats. Okay, so the main way in which the virus is transmitted is from blood-to-blood transmission, and the most efficient way in which the virus is transmitted is mother to child, and that's as the child passes down the birth canal, the child becomes infected. Also, horizontal transmission from person to person is an important transmission route, as is parenteral transmission, which is non-oral, and sexual transmission. Generally, perinatal transmission and horizontal transmission are the most common methods of how this virus is transmitted in developing countries. And in developed countries, what you find sexual transmission and parental transmission are the most common ways in which this virus is transmitted. And this is reflected in this graph, picture of the globe, showing you, according to the prevalence of hepatitis B surface antigen as set up by the World Health Organization. You can see high prevalence areas include the Arctic Rim, parts of South America into sub-Saharan Africa, and into the Far East. Within the European region, the prevalence on the whole decreases as you go from Northern Europe to Southern Europe. And overall incident rate varies from less than 1 to 15 per 100,000 cases. This gives rise to 90,000 chronic cases and 20,000 annual deaths. Now the UK is a low incidence and a low prevalence country. Prevalence is estimated to be 0.3%, and in relation to incidence, based on laboratory reports, we have a rate of just over 1 per 100,000 cases. Okay, this is data from England showing that the majority of acute infection is acquired in adulthood, 25 to 64 years, dominated, as you can see, by males, most of the infection in males compared to females. And so most of these groups here are occupied by high-risk groups. And so the UK is quite unique in the fact that infection is mainly acquired in adulthood. If we now look at active immunisation, that really now is the mainstay for primary prevention of this infection. And there are two main vaccines, plasma-derived vaccines and recombinant vaccines. Direct and indirect effects include that they obviously prevent infection, and they, as we've heard, they can increase herd immunity. And also what we should never forget about hepatitis B, it was the first vaccine developed that prevents cancer. So, plasma-derived vaccines, they were available first in 1982, and they are highly purified preparations of hepatitis B surface antigen from the plasma of asymptomatic chronic carriers. However, there were safety fears about this vaccine when it was put together because of the fear of using human plasma and the people acquiring HIV as a result of this vaccine. However, these vaccines have been used extensively, but in 1984, slightly later, recombinant vaccines became available, and these were using DNA recombinant technology. And in these instances, hepatitis B surface antigen was expressed in yeast cells to produce recombinant subunit vaccines. And these vaccines are licensed for use in the UK. So this is saying plasma-derived vaccines are used, and they have been used extensively in a number of countries. One of these countries being Taiwan. We'll look at some of the data from Taiwan a bit later. So in relation to vaccine efficacy, protective efficacy is directly related to the development of antibody to hepatitis B surface antigen. And vaccine efficacy is known to be greater than 90%, and the vaccine is protective against all known genotypes and serotypes. Following vaccination, when levels of 10 million international units per mil are achieved, these are indicative of protection against clinical illness and acute and chronic infection. With respect to long-term protection, hepatitis B immunity does wane over time. However, it is clear from some of the booster dose studies that have been carried out that vaccinees, when challenged with the antigen in the form of vaccine, most vaccinees will show immunological response. So for this reason, booster doses are not recommended for people who show an adequate response to primary hepatitis B vaccination. However, in the UK, our guidance does stipulate that healthcare workers should receive a single booster dose of hepatitis B vaccine 5 years after the initial primary vaccination. Okay, this shows you some of the direct effect of hepatitis B vaccine in England and Wales. This is data in relation from the early mid-80s up to the late 90s in healthcare workers, acute infection of hepatitis B. OAI stands for occupational acquired infection. We see this decline in the incidence of hepatitis B in healthcare workers due to the introduction of hepatitis B vaccine. Also, this is data from Taiwan. Their universal vaccination was introduced in 1984, and this has had a direct effect on the incidence of liver cancer deaths in children. We see this decline in the rate amongst children in Taiwan. And their campaign has been one of the longest in existence. So, in relation to the UK policy, we have a selective vaccination policy in the UK. People who are at high risk of infection are targeted with HepB vaccine. We also have antenatal screening to identify antenatal women, and so therefore children born to these women can be given vaccine. There are a number of additional policy options. They include vaccination of adolescents, vaccination of infants, universal vaccination, or a geographical selective policy, not too dissimilar from the BCG vaccination at present, or a combination of any of these. So, if universal vaccination was introduced in this country, how effective would it be? Well, an adolescent policy aimed at 11 to 12-year-olds would have no impact on perinatal transmission. Perinatal transmission would still occur, and so you still have to carry out antenatal screening and provide a vaccine for children born to positive mothers. If an infant vaccination program was introduced, obviously that would have to be delivered at the same time as DTP, at 2, 3 or 4 months. Again, that would still not prevent perinatal transmission. Antenatal screening would have to continue, and also selective vaccination, because in this country, hepatitis B is mainly acquired in adulthood. So these are the groups as clearly outlined in Immunisation Against Infectious Disease, who should be targeted and given hepatitis B vaccine, including injecting drug users, people who inject drugs, people who change sexual partners frequently. RAI stands for residential accommodation for those with severe learning disabilities, and travellers and prisoners. So these are the groups who should be vaccinated against hepatitis B infection. So, in relation to children that are born to positive mothers, this is a post-exposure prophylaxis in action. The vaccine can also be used for post-exposure prophylaxis in these situations for individuals who receive percutaneous injury, such as bites, a needlestick injury, and also sexual contact. So, vaccine and immunoglobulin can be used together to confer both active and passive immunity. So this is a close-up of the infant immunisation schedule. This is screening here, antenatal screening, and if a pregnant mother is found to be hepatitis B surface antigen positive, and the antibody status is E antigen is positive, antibody is positive, then the child is given vaccine. The first dose to be given within 24 hours of the child being born, and then at 1, 2, and 12 months post-birth. And here is our child born to antenatal positive woman, and the mother is found to be anti-HBe negative, or the E status is not known. The child should be given vaccine within the first 24 hours of birth, alongside immunoglobulin as well. And then again, a dose of vaccine at 1, 2 and 12 months post-birth. So this is the accelerated schedule, 0, 1, 2 and 12 months. And at 12 months, serum sample should be taken from the child and tested for the presence of hepatitis B surface antigen. This will exclude infection, and it will identify vaccine failures. And women that are found to be positive should be referred to liver specialists. Okay, now the World Health Organization has recommended that all countries should introduce universal vaccination. Many countries have done this. They actually also recommended that if the prevalence of HepB in your country was less than 2%, an adolescent program could be introduced in addition or in place of an infant vaccination program. The United Kingdom hasn't done so. It doesn't appear to be cost effective to introduce such a program. But a number of European countries have either adolescent or infant immunisation programs in place at the moment. Now, 2 recent countries, Netherlands was the last country, they just recently introduced universal infant immunisation, and the Republic of Ireland have had universal infant immunisation in place since 2008. And they found that it was more the economic analysis showed it's more cost effective to use universal infant immunisation compared to selective immunisation using a six component vaccine: DTP, HIB, Haemophilus, polio, and hepatitis B. So this now has been in place since 2008. However, they still have to continue antenatal screening and identifying children born to positive mothers and selective targeting of high-risk groups. However, in the UK, a clinical trial will be taking place very shortly. And this is a phase 4 trial looking really at the primary and booster dose response to a 6-in-1 vaccine. Infanrix, which is used in a number of countries. I know Australia uses it. I think they may be using this in Ireland as well. And this will be delivered using a 3-dose schedule, 2, 3, 4 months to coincide with how DTP is delivered in the UK. The main primary objective is to look at the HIB antibody responses, because there is a fear of HIB resurgence. So, as Andrew mentioned earlier, it is likely that HepB will be introduced universally in this country, but at present I'm not able to put a time point on it, but we'll be following this particular study. Now, these are figures from the World Health Organization showing you countries which have a universal infant immunisation program in place. We can see that the number of countries that have introduced such a program have increased, 180 now, and coverage, which is measured by the 3rd dose, is approaching 80%. And if we look at the globe, we can see areas which have coverage approaching 90% are in blue: North America, parts of South America, and into mainland Europe and the former Soviet states. So just to summarise, HepB does have a huge burden, causes a huge burden worldwide, and that's there are 1 million deaths. However, there is effective control and effective ways to prevent this infection, and universal and selective programs can be used to carry out this prevention and control. Universal infant immunisation now is really the mainstay of the prevention of hepatitis B worldwide. If programs are going to be introduced in those remaining countries that aren't vaccinated, and also for ongoing monitoring of systems, it is apparent that we do need good surveillance data and good economical analysis to give us an idea of what is actually taking place as these programs are being introduced. However, what is clear and what is apparent is that there is a huge potential then for HBV to be eventually eradicated by global immunisation. So there is a lot of potential that this virus can be eradicated from the world. So finally, I'd just like to thank my colleagues and acknowledge the colleagues from the HBA who I've spoken to and got some information from the related part of this study. Thank you very much.